Thank you for your participation. TOYO Corporation's agarose gel microcapsule, or AGM, reagent kit is now available. This video shows the progress of development of the products as of June 2023. Agarose gel microcapsule, or AGM, technology allows a single cell to be embedded within an alginate core and an agarose gel shell. AGMs are compatible with an aqueous environment and provide a favorable capsule environment for DNA amplification and cell cultures. This is Reagent's patented technology. Our reagent kit, the AGM Reagent Kit, offers the patented technology. I would like to discuss the structure of AGM in detail. The capsule has a core shell structure, which means that a liquid alginate core is contained within an agarose gel shell. The video shows that an embedded E. coli cell moves randomly in the liquid alginate core. The movement is known as Brownian motion. Since the shell or outer membrane is made of agarose, the material forms a mesh-like boundary that allows enzymes and reagents to pass through the pores as shown in the electron micrograph in. The photo is an electron microscope image of a microcapsule. Although the photo represents its dry condition because of the associated technical imaging limitation, it generally has a pore size between 200 to 400 nanometers. If the microcapsules are prepared according to the currently proposed protocol, they have diameters between approximately 20 and 100 micrometers, and the diameters are normally distributed with the most common value of 50 micrometers. The estimated size distribution may be changed to some extent if the stirring technique is adapted. Appropriate mesh may be used to extract microcapsules with any specific capsule size. The technologies related to preparing and using AGMs have already been granted intellectual property rights in Japan, and an international patent application was filed under the PCT to protect their IP. Papers on the technologies have been published in some major international journals, and press releases have also been issued. The slide represents substances that move within and through the AGM when subjected to the MDA reaction, which is expected to be the most common use for the technology for the moment. AGM shell is permeable to enzymes and substrates, but not to amplified DNA, other genomic DNA, or microorganisms. The selective permeability is also useful for preventing contamination. The core has as small a capacity as several tens of picoliter, and the tiny size helps reduce amplification bias when a single cell is subjected to the MDA reaction. TOYO Corporation marketed the reagent kits for making AGM last January. We provide a protocol for embedding single cells using only common equipment, such as the vortex mixer and centrifuge. One of the main uses of AGMs is DNA amplification before single-cell genomic sequencing. AGMs effectively reduce amplification bias during the amplification. The slide represents a flowchart of single-cell genomic sequencing using an AGM. The right section lists typical reagents used in the steps. The left section indicates the steps for genomic sequencing. AGM, our product, is engaged in the first step or encapsulating a single microbial cell. I would like to discuss what might be delivered by actually using the kit. The lower left image represents an AGM with one E. coli cell embedded, and the lower right image represents DNA amplified using multiple displacement amplification or MDA reaction. The DNA was stained with cyber green. Although MDA reagents and cyber green are not supplied with the kit, this is a typical output that the product may deliver. I would like to talk about the background to the development. A variety of microorganisms exist in our environment and are deeply involved in human health, the environment, and industry. For example, in terms of health, it affects the intestinal environment, resolution of pressure sores in the elderly, and exacerbation of atopic dermatitis. As for the environment, termite intestinal bacteria function as cellulose decomposers in biomass circulation. Since 99% of these environmental microorganisms are difficult to cultivate, their functions have been unknown until now. However, in recent years, aggressive efforts to understand the functions of the microorganisms have been pursued through genomic DNA sequencing. The sequencing methods include metagenomics, which are used for analysis of a collective genome of the microbial population in the sample, and single-cell genome analysis, which focuses on isolated single cells for individual genomic sequencing. Since metagenomic analysis involves a process of assembling recovered base sequences on a computer, the technique has several challenges. Some sequencings fail to recover full-length genomes, some sequencing multiple genomes may be mistakenly combined into one, or minor microorganisms are likely to pose difficulty in detecting them. On the other hand, the single-cell genome analysis technique, which involves isolated microbial single cells, is used for complementary purposes, including creating reference sequences for metagenomics. However, Since the technique includes the step of amplifying a small amount of genomic DNA using the multiple displacement amplification, or MDA, technique, localized DNA amplification may cause amplification bias and makes it difficult to recover entire genomic DNAs. The difficulty was the long-standing challenge for the technique. The slide lists the advantages of the AGM technology. Since it allows for genomic sequencing for a single cell, the resulting information is highly accurate and effectively complements metagenomic sequencing. The sequencing, therefore, delivers genome information of individual species, genomic analysis of unknown species, and highly accurate genome information. However, the conventional single-cell sequencing had some challenges: limited amount of DNA and accuracy reduced by amplification bias. AGM technology was developed in order to address the challenges. The slide showed how the challenges were addressed. We embedded one cell in a picoliter scale capsule to minimize reaction volume and reduce amplification bias. For example, we achieved genome coverage of over 90% for E. coli. The slide is also available on our website on AGM Reagent Kit. The text is reproduced from a paper. Blue areas represent the conventional single-cell technique. After cells were isolated onto PCR plates using FACS, they were subjected to MDA reaction. Red area represents a single cell subjected to the MDA reaction within an AGM. The data indicates that the genome coverage was significantly higher. In summary, the AGM reagent kit has three important features. First, small sample volume required; second, high genome coverage rate; and finally, low cost and high convenience. Especially, regarding the third point, we have in place a protocol that does not require expensive specialized equipment unlike other technologies, and it would facilitate many laboratories to try the single-cell genomic sequencing. The slide summarizes the advantages of AGM from different perspectives. As for the time required for the embedding operation, approximately 5 hours are sufficient to complete the embedding step if the reagents are used according to the protocol. As mentioned later, if the droplet generator, equipment developed by On-Chip Biotechnologies, is used in the automated manufacturing process, the time may be further reduced. Only a set of basic operations, including stirring, mixing, washing, and centrifugation, are required for preparation. No expensive specialized equipment is required. AGMs individually have a sol shell structure. Alginate sol contains a single cell, and the sol is encapsulated in an agarose shell. The AGM materials, alginate sol and agarose, allow ingredients to move through the capsules and support buffer exchange and enzyme reactions inside AGMs. AGMs achieved 90% genome coverage. The technology is a promising tool for single-cell whole genome sequencing. AGMs are storable in aqueous buffer. Since only a small amount of sample is required, the technology may be useful for uncultured microorganisms. The slide lists the advantages of AGM technology over conventional single-cell embedding techniques. Let's compare the conventional and AGM techniques using a specific example. I will compare the AGM technique with other gel beads and water and oil droplet techniques. Like gel beads techniques, the AGM technique allows a buffer and reagent exchange. However, cells more easily diffuse and are, therefore, more amplified in the microcapsule than in the gel beads. Indeed, a single round of genome amplification is sufficient to provide the amount needed for DNA sequencing. AGM has the advantages of lower amplification bias and therefore higher genome coverage. Also, cells are more easily isolated in AGMs than in water and oil droplets. Since AGM technique does not require expensive specialized equipment and is conveniently applied, it may be easily used in many laboratories. It is another advantage. On the other hand, AGMs do not have uniform capsule sizes, therefore, we position the AGM technique among other complementary technologies. We hope that customers are aware of its complementary nature. The AGM technology was invented by Professor Hiroyoshi Aoki when he worked at the National Research and Development Agency RIKEN. His paper was accepted as a scientific report by the journal Nature in 2022. As the developer, Mr. Aoki joined us in April 2023 and is now committed to customer support and development. His story about the product development are published in the technical magazine on our corporate website. Please visit the site if you find it interesting. The flowchart is the same as provided earlier. I will explain the steps from AGM production to pickup. The first step is the principle of embedding a single cell. The step includes two processes: a process where the sample is emulsified with alginate and gelled to contain it in the core, and another process where the core is embedded in a shell and then solubilized with a chelating agent. Both processes are based on a set of basic operations starting from stirring as shown in the slide. According to our own rules of thumb on the number of successfully embedded cells, you should have 3 by 106 cells, that is, 3 million cells, ready. And you will theoretically obtain approximately 300,000 AGMs with a single cell embedded. Earlier, I mentioned that the embedding operation may be completed in 5 hours. It is, however, only the case when a set of the processes are continuously completed. If you take breaks and put some time between processes, each process will take approximately 2 hours to complete. The manuals are available in Japanese and English. They are downloadable at our website on the products. Anyone can download them even if you have not purchased the product. If you are interested, please feel free to download them. In addition to the manuals, the MDA reaction protocols are also available as application notes. Although the MDA reagents are not supplied with the kit, the protocol refers to QIAGEN reagents, the most common reagents. The next step is the isolation process. After the MDA reaction, pick the AGM you want to subject to sequencing. Stain AGMs with cyber green and find ones that strongly fluoresce green under a fluorescence microscope. Otherwise, add cyber green in a higher concentration than usual and pick AGMs that appear orange under white light because some fluorescence microscopes have an upper limit to the fluorescence intensity. Manually prepared AGMs have to be picked up by again manually sucking them up with a micromanipulator or pipeman. Although the current procedures substantially depend on those manual operations, we will explore techniques that would require fewer manual operations, including limiting the dilution technique for cell isolation. In addition, if you use the droplet generator, made by on-chip biotechnologies, which I earlier mentioned briefly, to automatically create microcapsules, the capsules will have more uniform capsule sizes. They are expected to be more compatible with a selector or cell sorter and to achieve higher throughput. The photos show the workflow of picking up a microcapsule using a micromanipulator. You may pick 15 to 30 samples in a day with the technique, depending on your proficiency with the technique. We are always available for inquiries including requests for picking support. Please feel free to contact us at our website on the product. We will explain the automated preparation of AGMs. The AGM technique has the advantage of preparing the microcapsules without using expensive equipment. You can prepare AGMs manually and at low cost. However, the difficulty in making capsule sizes uniform and the need to manually pick up AGMs pose challenges to achieving higher throughput. The on-chip droplet generator and droplet selector, which are products of on-chip biotechnologies, will solve the problem. The droplet generator is capable of producing approximately 1,000 droplets with size of 30 micrometers per second, reducing the time required for the process, which would take 5 hours manually, to approximately 1.5 hours. The photo on the left shows how the on-chip droplet generator, made by on-chip biotechnologies, prepares AGMs. Similarly, the photo on the right shows how the on-chip droplet selector performs selection and single plating. A protocol for preparing AGMs using the on-chip droplet generator is also available. At the moment, the current protocol is intended to prepare products with a size of 60 micrometers. Now, however, we are considering preparing protocols for other capsule sizes. The protocols are also downloadable at our website. Before closing my presentation, I will provide data about the use of AGMs for culturing cells, which is another promising use for AGM in addition to single-cell genome sequencing. The photos, taken before the kit was made commercially available, show that a cell is cultured within an AGM. The photo on the left shows an AGM with one cell embedded in it, and the photo on the right shows that cells grew within the same microcapsule. Both cells were stained with cyber green. There are 2 precautions for culturing cells with the kit. First, you should prepare AGMs on the same day when the core is prepared. Data obtained indicate that the growth rate remarkably decreases as time elapses after preparing the core liquid. Second, you should replace EDTA, which is reagent number 10 in the kit, with another reagent as silation additive to alginate in the process of preparing AGMs. You may use, for example, 0.5 m sodium citrate buffer and 1% sodium chloride as the replacement reagent. It is because EDTA may damage cells. Keeping the precautions in mind, we embedded E. coli using the kit and cultured it statically at 37 degrees Celsius for 1 day in a 15 ml tube containing 20 L of AGM and 1 ml of pound medium. The results showed that E. coli grew within the AGM. Although only an experience with culturing E. coli is available, AGM technique is a promising option for culturing a single microbial cell. A protocol for cell culturing is also available. Like the protocol for MDA reaction, It is downloadable as an application note at the product website. The slide shows our vision for developing additional AGM reagent kits. The AGM kit currently available is designed for use with microorganisms. It represents the first step for addressing unmet needs with AGM technology. As a second step, we are working on developing a reagent kit for human and mammalian cells. The reagents supplied in the currently available kit cannot be applied to human and mammalian cells because of their pH and osmotic pressures. The reagent composition of the new kit is currently almost complete, and we are currently working on developing a kit. Our efforts to obtain consumer feedback and explore unaddressed applications are ongoing. We would appreciate your input. The slide lists frequently asked questions. We have received more inquiries. We plan to set up a FAQ section on the website on the products later. Although it is supposed that AGMs will not preserve RNA and proteins, we are currently working on possible methods of preserving those materials. One AGM reagent kit can support 3 reactions. It can be ordered from your regular suppliers. If you have any questions, please contact the sales representative via our website on products. Now, I close my presentation on the AGM reagent kit. Thank you for your attention.